proceed with the last two talks by Franco Cecchi, Professor of Cardiology in the University of Florence. Franco, keep as much the time as you can. You are the last speaker. You have two talks. Maybe you can merge as much as you can. Franco.
Well, first of all, let me thank Yorgos Eftimiadis for the kind invitation. And I really regret not being able of being with you. Can you see my slides?
We can see one slide of your face, which is wonderful.
Yes. Okay. All right. Again. All right. So, okay. So, well, you know, it's I really regret not being there because, you know, I believe that, you know, I have really many friends in the room. And I believe that this topic is really a very important topic right now. Now, gene therapy for patients with hypertrophic cardiomyopathy. And actually, let me just add, is the...
Sorry, we cannot see your slides.
You cannot see the slides, but I was actually sharing.
Yes.
Now, okay, now it's okay.
Okay.
Okay, all right, sorry. Okay, can you see then?
Yes, yes, carry on.
Okay, so dream or reality? And let me tell you that, I mean, this is an important question because, you know, with these big advances, I mean, this will be probably the greatest one, I mean, if it will happen in the future. So let me just tell you that, I mean, we know really a lot now about hypertrophic cardiomyopathy. And just to share with you this, you know, one of our latest papers that really collected data on the original cohort that we saw in Florence with an average time of 27 years up to 50 years. And we all know that, you know, what the disease is in general. I mean, what is the risk of sudden cardiac death? What is the risk of heart failure? And also at the same time, I mean, how much we got, I mean, comparing what we actually observed in our original cohort, the first 10 years, and then later when more therapy and more we understood, you know, better how to treat this patient. We have a big armamentarium actually right now. I mean, the big armamentarium also got finally new drugs that really act on the real cause of the cardiomyopathy failure, both hypertrophic with obstruction or non-obstruction and dilated cardiomyopathy. So we have the myosin modulators beyond all the procedures and you know, drugs and all guidelines that we all know and we are all aware of. So this is really very important. As you know, it is very important to understand, and we all know about that, you know, we have a gene cause in, you know, about 50% of the patients. Then we have several other diseases that, of course, have to be distinguished. And then we have, that's a process that we need to do in each patient to understand what is really the cause of that phenotype. And also we try, you know, right now we also start to understand also the other, I mean, the minority of patients that have no gene variant detected, but also we have some data which we don't fully understand yet why they do develop the hypertrophic cardiomyopathic phenotype. But this all is important for gene therapy because, you know, we are all aware of most common genes, of rare genes, which we heard also this morning, and also of the other disease which have a genetic cause. So this is really important in, you know, precision medicine and in really addressing the correct therapy in our patients. So coming to gene therapy at this point, I mean, I believe that gene therapy is really an emerging technique for the treatment of monogenic disorders. And so far, there are always many diseases, not only heart diseases, that are currently treated by this kind of therapy. Okay, what happens, you know, when you plan to have gene therapy? Well, you know, gene therapy aims to provide sufficient gene expression in cardiomyocytes to ameliorate or correct the disease phenotype. And, you know, as we shall see, several clinical trials are ongoing with promising preliminary results, both in animals and sometimes also in humans. Now, just to summarize, the very rare diseases, Danon disease, Fabry disease, Friedreich ataxia, you see, these are the open-label trials that are going on, and I will not be talking about that. But of course, these are limited to few people, and this will be very important also that could show us, I mean, how really we can solve the problems of these unfortunate people. And coming to other, I mean, you have the muscular dystrophy, the dystrophies, you know, like Duchenne, Pompe disease, and as well as TTR amyloidosis, which we understood how to diagnose and treat only in the latest 15 years. So this is really current therapy, current trials that are going in the future, I mean, to treat these people more probably, I mean, to give them a better treatment. But coming to hypertrophic cardiomyopathy of genetic origin, I mean, let me show that very recently, and Bill McKenna has already mentioned that, the first patient was enrolled in the phase 1b trial of treating the gene of MYBPC3. And why is this important? Because MYBPC3 is the most common gene that causes hypertrophic cardiomyopathy, sarcomeric hypertrophic cardiomyopathy. And at the same time, Thiene, this investigation company, also had clearance by the Food and Drug Administration to start testing gene therapy for the treatment of arrhythmogenic right ventricular cardiomyopathy due to PKP2 gene variant. Now, let's come and let me show a little bit shortly, you know, how this will work. I mean, and this is reported in a very latest paper published by our group, Alessia Giraud, who really is involved in gene therapy advancement. And you see that, you know, you may have two kinds of, simplifying, two kinds of problems. You know, you have a loss of function mutation or you have a gain of function mutation. And in this case, function, I'm really sorry because there is a lot of noise because somebody is drilling in the apartment next to mine. However, loss of function mutation, will produce either the absence of a protein or a partially or totally unfunctional protein. In the gain of function scheme, instead, you have a production of protein with increase of different activity or a production of protein that blocks normal function of wild type. And gene therapy at this point is defined by the introduction of new genes or the genetic modification of existing genes or their regulatory portion via gene replacement and gene editing strategies. And as you can see, in the loss of function mutation, you have the gene replacement. So introduce a new portion of the gene, which will produce the correct protein that will work finally well. While in the gene editing, you have to introduce a gene editing procedure that will cut out the gene that is working not properly, and that can be corrected by the insertion of a new portion or the deletion of this portion. So this is basically the simplified mechanism. This has been carefully demonstrated in animals. As you can see here, for instance, this beautifully reported experiment in mice. This is a gene editing procedure where you can see that in mice, in treated mice, the level of thickness, wall thickness, or the level of fibrosis is normal in the sense that it's exactly the same as, wild type animals and not of the genotype, the gene variants animals, the knockout. And in the same experiment, you can see exactly again, this is again reporting about the fibrosis, the wall thickness. So this has been demonstrated in mice. And let's see what happens not in gene editing, but in gene replacement. And in this case, let's go back to the MYBPC3. You see that MYBPC3 is now and is necessary for normal structure and sarcomeric function. It modulates cardiac contractility and relaxation. And when you have loss of function variants, they cause MYBPC3 sarcomeric hypertrophic cardiomyopathy because you have low levels of MYBPC3, abnormal sarcomeric function, and you can see here the examples of histologic specimen in knockout mice, and this can be restored after the injection of the corrected gene. So there is in the gene therapy concept, you have the transgene, which is usually inserted through the infusion of the viral vector. I mean, you have the correction of the MYBPC3 cardiomyocytes, and then the transgene produces MYBPC3 protein necessary for normal cardiac function. And this is how it works. You have, the delivery of this capsid, which is going to be inside the adenovirus V9. Okay, then it's infused, it's received by the cardiomyocytes, and then the mutated gene, produce the corrected protein. And that's how it's working. The MYBPC3 transgene forms episomal DNA. It does not integrate into the host genome. The transgene encodes cardiac myosin-binding protein C, and there is a durable expression of MYBPC3 anticipated due to the fact that the cardiomyocytes do not divide. And these are the results. And as you can see here, I mean, this is the left ventricular mass in mice. And you see that the hypertrophy is reduced in comparison with the knockout animals, and they see that the survival is greatly increased. So you have that the durable FV mass and other functional parameters improve 1 year after the injection, and there is an increase in median life of the animals over 6 months, and heart failure and fibrosis markers improve after treatment. So this is the animal experiments. Now let's come to the trial, which has just started. And this is a safety and tolerability dose finding and pharmacodynamics trial, which has been approved and started. And these are the key eligibility criteria. You need to have a confirmed truncating MYBPC3 gene variant. These are all non-obstructive HCM patients. They must have an ICD. They must be symptomatic with NIA class 2 or 3. They must have at least a proBNP greater than 300, left ventricular ejection fraction greater than 50%, normal hepatic function, and that's a very important point. They need not to have neutralizing AAV9 antibodies. And this is a crucial point because, of course, these antibodies are present in a fair number of the population, including HCM patients. This is how it will work. This is the AAV9 viral vector infusion captured by the cardiomyocytes and production of the corrected protein. Well, is it all good? No, there are some caveats. Of course, some hepatotoxicity events and some uremic syndrome, hemolytic uremic syndrome have been reported. Cardiospecific low immunogenic vectors and adequate immunotherapy regimens are unmet needs, and this is still to be solved. And then there is another problem. When you start such trials, the small sample size, the necessity of certain points, and the lack of a control arm make designing clinical trials really challenging. However, I believe that in the next future, a dream might really become true. I thank you very much, and I'm sorry for the noise due to the drilling problems in the above flat. Thank you.
Very much. That was a great lecture. I think we should proceed to your second talk about Professor